Bleeding caused by traumatic injury is responsible for 1.5 million deaths globally per year, disproportionately affecting a young, working-age demographic. Additionally, bleeding is the leading cause of preventable death after injury. Current trauma care relies on wound dressings made with highly absorbent materials that soaks up and leaks blood. The soaked dressings stick to the wound and when removed, cause pain and secondary bleeding, which also increases the risk of infection. We have a solution. A range of wound dressings incorporating a superhydrophobic material using both physical and chemical mechanisms to control bleeding, restricting blood flow and accelerating clotting, as well as demonstrating exceptionally low wound adhesion and antimicrobial properties. We are initially targeting our devices at paramedics, doctors and nurses in pre-hospital and hospital emergency care settings in the US, where there is a large and growing number of patients every year. We have great in vitro and in vivo test results from the lab, and now we are looking to raise £700,000 to help us reach our next milestone, demonstrating efficacy in preclinical trials. With our technology and your investment, we will save lives. Thank you.
Hi, that's a very good elevator pitch. My question is you picked US as your market.
Yes.
You picked paramedics. Why is that?
So first for the US market, it's because it has a very large number of patients suffering the types of wounds that we're targeting. And it's also one of the only countries where the number of patients is rapidly increasing every year. And we're specifically targeting paramedics and the sort of early pre-hospital and early care periods because stopping bleeding as early on as possible is more effective for more patient outcome and mortality.
Okay, sorry. So you mentioned some in vitro in vivo test results in lab, I assume, right? So can you share any data on that efficacy? I know there's no clinical trial yet.
Yes. So so we've done testing on clotting speeds and we've demonstrated that it's faster than a control group with standard gauze. We've done testing with adhesion force, which has shown, I believe, 20 to 50 times less peeling force than competing products. And a little bit of in vivo testing for looking at toxicity and just making sure it's harmless.
I'll just follow up a little bit on your preclinical data. If I recall a long while ago, things like Roman effect, which is the kind of absorption of proteins onto surfaces over a period of time, can catch you out. So how long have you been looking when you're doing your adhesion tests and your clotting tests? Over what period of time are you working?
I believe it's over no more than a range of minutes, 5-10 minutes. Yeah.
And then as you move to preclinical, I'll say 700K feels like an ambitious number. So what are you targeting to do with that 700K and how's that going to move you to the next kind of value inflection point where you can get 10 times that for next stage preclinical?
So a large chunk of that will go towards operating costs for 2 years, so about 400,000. And then there will be about £100,000 for the preclinical trials where we'll be, again, as I said, testing for efficacy and safety. And then additional money for consultancy fees, regulatory applications, healthcare economics, and development costs to help us achieve a sort of minimum viable products to take forwards to clinical testing.
Okay.